So I wasn't even recording yet. But so test 1 is over, but not forgotten, right? If you read my announcement. And the person that was sick is going to take it Friday morning. So once she does, I'll make it available, and you guys will be able to see your individual results. And do that as soon as possible, right? So Friday or this weekend, look and see, where you went wrong, right? And I'll talk a little bit more about the test on Tuesday. The average, I think, was almost a B, which was good. But there were some low scores, right? So there's some of you guys, and some of you guys that spoke to me even right after taking the test were like, there's still lots of room for improvement, right? But depending on how low, you may have knocked yourself out of A range, right? But B might still be attainable for this course. The important thing is learning from your mistakes for the final exam, right? Because this stuff doesn't go away. You're going to see some of it again on the final. So it's important to know where you went wrong, how you went wrong, how to improve for the next test, but also how to make sure you understand that material for the final exam, which is comprehensive. Make sense? Okay. So I'll let you guys know. I'll post an announcement, as I said, when the test results are available. Remember, you need the lockdown browser to be able to view those results, though. So either you downloaded it on your computer via the link I gave you guys here, or you can go upstairs to the Open Lab. They have the program. So remember, it's the lockdown browser program. And then the Hibernia Center where we took the test, right? If there isn't a class in there like we were hogging it up the other day, then you're welcome to go in there as well. And as you already know, they have the program on their computers in there. So today, try to remember to hit the right button. We're starting Module 2, right? And you'll notice I've reorganized the way I'm keeping stuff because the other way I didn't like so much. So all our objectives and PowerPoints are listed here on this page. Really? I hate that. My nose wanted to sneeze and it decided not to. That tingly feeling and nothing happens. All right, so all the objectives are already here, right? So these are all the topics that we're going to cover in this module. This is a virus model. This is pretty cool. You can download this, or I think you can just open it, but I don't know if you can. I don't think you can print from here. Yeah, they don't have like a print button. But so you'd want to download it and print it. And if you cut it out and fold it up, it makes the dengue virus capsid. It's helical. But I have examples of this. If you've been in lab, you've seen probably the poster board I have in there. But Tuesday I'm actually going to bring the poster board and the models in so you guys can see three-dimensionally what these isohedral virus protein coats look like. But this one is specifically, and you can search on the internet too. There's a whole bunch of other virus models you can make for fun. And actually, I don't know, was it still on the desk when you guys signed in for the test? Did anybody see it? I had given them a virus, it's a little scientific joke because, computers get viruses, right? I hope you don't get a virus. I hope this paper model is the only virus you ever see. Right? So, and so then here's our link for the lecture, and then, later I'll post my link that doesn't have anything missing, right? Has the tables filled out and everything like that. And then on the other page that I have, right here, are animations and videos for Module 2. So the other thing I asked you guys to do was do that Connect bonus assignment ahead of time, which was most of basically all these animations, but with questions associated with it. So to get you guys familiar about how viruses work and all of that before I even lecture on it. Because I know a lot of you guys don't like to read. Right? But you usually like the animations. So these are links to the animations, all of these, and then there's even some videos of me in lab and lab stuff too, as it relates to the growth unit. So I have that all in one spot, and then I will add recorded lectures to this page that says recorded lectures. You know, there'll be links all down the page as we go. And what I'm going to do, and I'll show you, I'll start my page. What I'm going to do is today is the 18th, so I'll even date them. So that way if you're absent, it'll help you like keep track of what you missed. So today is going to be viruses part 1. And so the video will be right here later. So the other thing I asked you guys to take a look at ahead of time, which I even did my homework last night, I looked at it, is one of the case studies. So hepatitis is a virus, right? And so this is the actual case study from a case study book that I scanned. So it's about, well, you tell me. Anyone read it? What happened? Yeah, she got hepatitis from onions at a restaurant. Man, date night really did not turn out so great, huh? Yeah. So she got this viral infection from food that had been contaminated. And there are lots of different viruses, right? If you look at, and for each one of the case studies, other than the Listeria one, should have put this one towards the top. There is a PowerPoint that goes along with it, right? So if you open up the PowerPoint, it even has the case study embedded in it and the questions, and has most of the answers. Here, I can make this, can I make this full screen? Yeah. All right, so here's the case study, right? And so for hepatitis, right, there are currently, and I think there's actually more than this. I think it goes past G now. Yeah. So you'll notice that this table is really good because it has a lot of really important information in it. And so for her, she had hepatitis A, and this is commonly transmitted fecal-oral. So people get the infection, right? It's it can be transmitted through their digestive tract, and then someone else come in contact with their poop, basically, orally, and it enters into their system. So what happened is that somehow, somewhere in the process, right, the green onions that she ate got contaminated with basically somebody who had the infection's feces, right? So, and that's one of the things that this case study talks about too, is like, you know, is finding that source and educating those sources. So the scary thing, y'all, is I lived in California in the salad bowl region for a little while, in the Salinas Valley, where a lot of, they call it the salad bowl because a lot of the things that go into salads are grown there. You pass by fields upon fields upon fields. There's no bathrooms out there, y'all. The people that are out there picking that food, right? Yeah, they're going to the bathroom in the field. So now there are laws in place that they have to have portlets out there, or portlets on the trucks as they come in with the field workers, so that they can go to the bathroom in a safe manner so they're not contaminating the crops, right? These, you know, this is one of those things that you're like, we kind of should have thought of this like a long time ago. But, you know, think about when stuff comes from other countries. Do they have that level of regulation that we have gotten to now in the United States? Or whether they actually follow it, yes. Right. So, I can't remember, did they tell you where it came from in this case study? They came from Mexico? Yeah. So, I mean, it came out of the country, too. So, I don't know about you guys, but when it comes to my produce, if it doesn't say USA, I kind of, and I try and stay local too, because then it's fresher, right? And probably a little bit better choice. So the other thing I wanted to point out here too is the genome. So most of these guys are RNA viruses, which means their nucleic acid is RNA, right? This is one of the things that we're going to talk about today. One of them, though, the hepatitis B virus, is DNA. So there's one distinctive difference there. And then if you'll notice, too, though, hepatitis D is not a virus. It's a viceroid. It's an RNA that gets inside the hepatitis B virus. So the good news nowadays, too, as it relates to hepatitis A and hepatitis B, what do we have to help protect us? We've got vaccines. And because D is found inside of B sometimes, the vaccine for hepatitis B will protect you as well against the viceroid known as hepatitis D.
So we've taken out 3 of the 6 or 7.
Yeah. Yes, with vaccination. And so one of the things that I posted for you guys in this area for this case study too, I wish you could, I guess I could hit the back button, right? That would make sense. Still learning navigation and how to best set things up in Canvas. So the other 2 things I have posted for you guys here are the information sheets from the CDC, from the Centers of Disease Control. Basically, when you go get a vaccine, what they give you. So this is it. And of course, I want you to read it. So I asked you questions about it in the case study, which I did not answer in the PowerPoint. But if you listen to my recorded lectures, I do answer all the questions. It's under the module. So notice all these documents under hepatitis, all of these you should open, look at, and read, right? And maybe even print out, because you can have these notes, right? Write on them, highlight them, and organize them when you take the test on all these case studies at home. It's a take-home test. But you can only open it once, and you can only do it once. And you have all semester to get it done. You got to get it done by like May 2nd, otherwise it doesn't go into your grade. So, the only one that has a quiz is the Listeria case study. So notice right here I've posted instructions for case studies. So, for the Listeria case study only, you will submit a quiz on questions I have posted from the case study. So I picked out specific questions I wanted you guys to look up, right, and read and figure out on your own. The other 5 case studies, including this hepatitis one we're kind of talking about right now, because it'll help give us a foundation for our virus lecture. I've given you the answers to the questions in the case study via the PowerPoint lecture, right, that I opened up and we were just looking at. And right here, notice I have recorded lectures. So this one actually has meningitis, hepatitis, Shigella, and actually Giardia, but I also have a separate one for Giardia and fungi. So, these are my usual YouTube lectures, so you can click on it, you can fast forward, right, and get to the particular one you may want to listen to, or sit down, have your notes, right, and write on it. So if you listen to that, it's going to tell you the answers, right, but you should read over the case study, right, if you haven't already. And then for each one of them, so you don't even have to look it up in your book, I have scanned a copy of either your book or another book that does a good job of talking about the particular disease So everything you need is right there, right? So there's hepatitis viruses. You probably want to print this one out to read it better, though. But it's also in your book, right? So just look up hepatitis viruses in your book. Notice, again, that's the table I have in my, no, that's not it, different one. That's the table I have right in the PowerPoint lecture, right? I took it right from the book. So I'm going to refer to this one, right, today as we're talking about it. And as you read through, right, it makes sense why the doctor asked her those questions. Had she traveled, right? Did she use intravenous drugs, right? She a drug user, right? And I don't know. It's not funny. It's actually sad. But you know how some of us, we laugh when things aren't funny, right? It's a nervous reaction to certain things, right? For some people. So if they're laughing, it doesn't necessarily mean they're laughing because they think something's funny. It actually might be because they're nervous, right? And I'm one of those people that happens to me. See? There you go. Just FYI. Or when something is concerning. So, when you read over these two documents about the vaccines that we now have and who we give them to and why we give them and what age and how many doses and who should get it and who shouldn't, right? It really gives you a perspective. And when I was reading over it last night, one of them was IV drug users should get this vaccine. And I kind of laugh because do you really think that people that are using illegal drugs think about these kinds of things? Oh, yeah, I should go get vaccinated? No, they're thinking about when's the next heroin dose coming, right? It's kind of sad. This is why, too, I feel privileged to be able to sit here and educate, right? Because that's the most powerful thing that we can do, is educate others about these things, right? If they knew, right, maybe some of these things could be prevented, right? This is the most powerful thing for us, is education. Knowing about these things, right? Being able to tell others about these things, right, to help protect them. So, hepatitis, I'm not going to answer the questions for you. Answer them, we'll talk about it next time. We'll talk about the vaccines next time. Because I want you guys to actually read this stuff. I actually want you to read it. So, for all the case studies, you'll see this. You have the case study, right? You may have some supplement information. In this case, I gave you 2 textbooks to read from, and then the case study itself. And most of the questions are answered in that PowerPoint, in that case study PowerPoint, but some of them I don't, right? You would have to actually listen to the recording to get all the answers, okay? So Listeria is the one exception, right? You have questions, you have the case study, but these are the questions I really want you to know the answers to. Here's a book that you can read to get the answers. Here is an assignment, right? So once you open this, open it once, notice it's worth 20 points, right? You'll put in your answers, right? So answer those questions, and then you'll basically take a quiz based on those questions that you answered. You can have your notes right there in front of you. That's fine, right? Once you get done, it'll tell you the answers, okay? So that you know them for the test that you'll take on all 6 of these, which I didn't post yet. Or did I? No, I didn't. Because I really don't want any of you guys doing it yet. There's no way you're going to go through all 6 of those case studies this weekend. Maybe. I'll turn it on if you really want me to. If you want to get it knocked out now and be done with it, I'll turn it on. I'll turn it on tomorrow. If I don't, send me an email and I'll do it. Okay. Any questions on that? Right. And I'm going to, as we go throughout the semester too, I picked these particular case studies to go with what we're talking about, right? And especially for a lot of you guys that are going into health field type stuff. And then, like I said, is in general knowledge. It's good to know about these things, right? It's good stuff to know. Okay. So let's talk about viruses. So as far as infectious agents go, as we just saw with the hepatitis virus, right, we thought at one time one of those was, we thought hepatitis D, right, was actually a virus. Come to find out it is not. It's a viceroy. It's an RNA that inserts itself into the hepatitis B virus. So what are these things? What are viroids? What are viceroids? What are prions? So these are other infectious elements other than viruses that can make us sick. And all of these things, including viruses, are composed of nucleic acid. They can contain protein, or they may not even have nucleic acid. Who do they infect? Do they infect people? Do they infect plants? Do they infect bacteria? Right? Who's the host? What cell does this infectious agent get into? No. Depends. So, viruses, viroids, viceroids, and prions, these are all non-living elements. These are not cells. They're not organized to the cellular level. They're not, therefore, considered organisms. They usually only consist of a few molecules found in cells, right? So, nucleic acid or protein. They are obligate intracellular parasites, which means they must get into a cell in order to replicate themselves. They don't have the machinery or the ability to make copies of themselves on their own. Outside of the host cell, they're considered inactive. Right? So a virus outside of a cell is considered inactive. It still can be infectious though, right? But sometimes we can use certain cleaning agents that we're going to talk about later in this unit that can deactivate or destroy viruses so that they're no longer infectious. So if you come into contact with it, it can't get into your cells. It can't make you sick. Inactive means that it's not metabolizing, it's not doing anything. It's just there. Infectious means it can cause infection. So, as far as viruses are concerned, all of them have protein. So what inside of our cells is it using to make its proteins? What makes proteins inside of our cell? Ribosomes, right? Viruses don't have ribosomes. They use our ribosomes or whatever cell's ribosomes it gets inside of to make its proteins. The enzymes that we use to make DNA into RNA, they use those to do things like make RNA from their DNA. I'm in the process of trying to get us a new book for this course, right? Because this one is not so great to read, right? It's not cool, I know. So we're in the review process right now, and there's a picture that I'm going to steal from one of the books that I want to use because it's a really good one. You know, they say a picture is worth 1,000 words. Like this picture is worth 1,000 words. Like perfect. And so I'm going to steal it and have it for next lecture. I'm going to scan it tomorrow because I really like it. Because it really does a good job of summarizing the different types of viruses based on their nucleic acids and how they achieve their goals, right? How do they go from what they are to making themselves, using the machinery within our cells. So there's lots of different types of viruses. Like we already pointed out when I looked at the different hepatitis viruses, one of the things they had listed was RNA for a genome, but remember one of them is a DNA virus, and that's hepatitis B. And so that virus acts differently in our cells than the others that are RNA viruses. So I find, too, sometimes if we have a real-world example, too, right, kind of helps solidify this information. So that's one of the reasons why I asked you guys to look over that hepatitis case study. So as it applies to each of these infectious agents, viruses, as we've already said, do they have nucleic acid? Yes, because they're either DNA or RNA. And that's an important distinction there, is that they're either one or the other. They're not both, right? They either have DNA as their nucleic acid or they have RNA. And then it even goes a little more deeper than that we probably won't get to yet today. Viroids, which, has anybody ever heard of a viroid before you started reading this stuff? No. Right? And there's a reason for that, right? So what are they? What are they?
They only infect plants, right?
They only infect plants, and they're only made out of a nucleic acid. What nucleic acid? They're just RNA. And yeah, and the fact that they only infect plants is probably why you've never heard of them before. Now, in the reading, it seems like we can't seem to decide what we want to call the next one, right? If we want to call them satellites or viceroids. So, of course, I have both terms there. And it depends really what book you're reading, whether they refer to them as satellites or viceroids. And so in our book, I think it's viceroids, right? And the other book that I'm reviewing right now, it's satellites. And yet another one, they use both terms. So unfortunately, you got to know them both. So for viceroids, what are they made out of nucleic acid-wise? Only RNA. And so again, remember the hepatitis D, right, is a viceroid. It is just RNA. Prions or pyrons, do they contain any nucleic acid? No. No. They're just infectious proteins. So we're not going to get to them until the next row. Viruses, do they have protein? All viruses do. All viruses do. All viruses have nucleic acid and are surrounded by a protein coat. So that model that I showed you guys, right, that dengue virus model, those are proteins, right? And when you cut out that model and fold it up, it forms what we call an isohedral. That shape to the protein coat that surrounds and protects the nucleic acid, but also is going to what helps transmit that to our cells. Because the very first step in viral infection is being able to attach to the cell in which it's going to infect. How it attaches is usually its protein coat, or some viruses we're going to learn have an additional layer called an envelope. But proteins are very important components for viruses. And again, why they have to get into a cell, because only cells have ribosomes. Right? Only the cell can copy and make their proteins for them to form their protein coats. Now a viroid, it's RNA only. So does it have protein? No. No protein. What about viceroids or satellites? That would be no as well, because we said protein only. Only means that's it, right? Only is that's it. So what about prions? They're infectious proteins. They are only protein. That's it. So viruses, what do they infect? They infect us, right? Can plants get them? Yep. Yep, plants can get viruses. Can bacteria get viruses?
Yeah.
Yep. Can algal cells and fungal cells and protozoa? If it's a cell, the answer is
Everything?
yes. all living organisms, all living cells could be infected by a virus. But viruses are pretty picky, thank goodness, right? Pretty much animal viruses stick to animals. Plant viruses stick to plants. And this will make more sense as we look at how it is a virus infects the cell. As we already know, animal cells are different from plant cells, bacterial cells or algal cells or fungal cells. Because these cells are different, there are different viruses that attack them. But viruses in general infect all living cells known on Earth. Viroids, as we said earlier, the reason why you probably haven't heard of them, they infect what?
Plants.
Just plants. Or mainly plants. Although I have to say I don't know of them infecting any animals, but I'm sure there's probably one out there.
Cheers.
It's not a living organism, right? So they're not eating. But they are replicating. And that's the bad news here for these guys, right? Is that when they copy themselves, they're using our cells. They're using our resources. And this can be detrimental to a cell. And especially if you're a single-celled organism, this could lead to your death. Right? So viceroids, where we said the hepatitis D, right, is a viceroid. Where is it found?
Oh, the hepatitis D.
Yeah, hepatitis D. It's inside the hepatitis B virus. So remember when we looked at the chart, hepatitis B virus is a DNA virus, right? But sometimes when somebody has hepatitis B, inside that virus is also some RNA. That's the hepatitis D viceroid. So what happened is that scientists, right, found this hepatitis B that had the DNA for hepatitis D in it. I mean B, D and B, oh my God, right? And it had some RNA in it. So they thought it was a whole different virus, right? They're like, okay, so it's next on the list, so it's hepatitis D. Come to find out that it was, as I said, hepatitis B with this viceroid, with this extra piece of RNA inside of it. So not all hepatitis B viruses have the D viceroid inside of them. Just some of them. So in this case, a virus is basically being infected by something else, right? So human viruses, and we have the specific example that we just went over of hepatitis B being infected with the hepatitis D viceroid. So it is just RNA, but how it travels is it has to be inside a virus. This is how viceroids or satellites get from one place to another. They actually are inside of a virus. That virus does not need them, right? These guys are hijacking viruses. Make sense? So prions, they infect who? Animals and humans. And y'all, we really are animals, right? Yeah. We kind of put ourselves on this little pedestal. Right? But we are animals. So viroids, are unable to replicate itself because its RNA does not encode for gene products. It doesn't make anything. So remember, think about central dogma when we think about our cells, what happens in our cells, right? We go from DNA to RNA to protein. that protein is the gene product. The gene is the DNA, right? The information on how to make that product. A viroid is an RNA. But that RNA doesn't interact with the ribosome. It doesn't get made into a protein. They believe there may be a host, so inside that cell that they infected, remember plant cells, the DNA independent, which means it doesn't need DNA, right? So like in our cells, we have to go DNA to RNA, right? Well, this is the polymerase that does not need DNA. It can turn RNA, it can make copies of RNA. So it can take that RNA and make copies of it. It can go from RNA to RNA. So this is an enzyme that takes RNA and turns it into RNA. It's DNA independent, unlike what we're used to, right? Usually you got to go DNA to RNA. And the mechanism in which it happens is referred to as a rolling circle mechanism. You can search YouTube and watch videos on it. Animations, if you want to see it in action. So why is it bad news? Why does it cause disease? What's the big deal? So a foreign RNA got inside of a cell, and the cell copies it, and you have a whole bunch of these foreign RNAs. And the ribosome doesn't even bind to it and make anything out of it. So what's the big deal? Well, we just said, we make RNA, it interacts with ribosomes, and we make what from that? Proteins. And our cells do this all the time. So do plant cells, right? They got to make stuff to do stuff. RNA, double-stranded or single-stranded? Single-stranded, right? And that's so that the ribosome can read it, right? If you've got RNA, your RNA, and then you got some foreign RNA that's also single-stranded. If those two match, they can bind with each other, right? They match enough. And now you have what? Double-stranded RNA. Can a ribosome read double-stranded RNA? Nope. So you've just essentially what we refer to as RNA silenced whatever that viroid is binding to of the messenger RNA in that cell. So depending how important, right, you probably made that messenger RNA for a reason, right? You want the ribosomes to probably make some protein from that messenger RNA. But now this viroid has come in and basically said, nope, you can't make that one anymore. I'm going to silence it. I'm going to duct tape right over that mouth. That's kind of bad news for the cell, right? You are making that messenger RNA for a reason. You want the ribosome to read it. You want to make that protein. But every time you make the messenger RNA, this viroid attaches to it and silences it. Stops you from making that protein. So as you can imagine, this can be detrimental to the cell depending on how important that protein is that it keeps trying to make and it keeps getting silenced by the viroid.
Is there a chance that the RNAs don't bind?
Right.
So
There are times where it won't bind. It's only going to bind to stuff that it matches to. Right? So the problem is this causes disease in plants because it's probably binding to stuff that's important. Otherwise, it wouldn't cause a disease. It wouldn't cause a problem for that plant. Now, I'll tell you, that's the extent that I know and understand this stuff, and that's the extent you guys need to know and understand. But if you're curious, go for it. I personally don't have the curiosity or the time right now to know anything more about viroids. But it's kind of interesting phenomenon if you think about it, right? This silencing, right? Binding with the messenger RNA and stopping that cell from being able to utilize it. So the viroids themselves, I guess for stability reasons, will actually form a closed circle at times. But of course, this can open up and become single-stranded and attach to those messenger RNAs. So again, it can bond with itself. And so you'll get this structure where you see these kind of bulges here. Well, that's where it's not binding. These straight sections are where it's binding. And I know this isn't the best picture that I stole from the internet, but you can see here where they're matching up. I can't blow it up anymore. But you know, we know base pairing, right, for DNA and RNA. It's not all that different. There is bonding that can happen between two strands of RNA. Although typically it doesn't happen in our cells so that the ribosome can read it. But that's because the ribosome reads it with the aid of transfer RNAs. Remember, the transfer RNAs are what have the anticodon, the matching between the messenger RNA and the transfer RNA so that the correct amino acid gets put there. So it's similar to base pairing in DNA, right? Except we have that one nucleotide difference in RNA. So it's still G's with C's because you have both of those in RNA and DNA. But A and T bind with each other in DNA, right? Adenine and thymine. But there's no T's in RNA, right? So it's U and A, right? The T is switched out for the U, so it's U and A binding. And so if you could zoom in on this, you would see that base pairing, A's with U's and C's with G's. So moving on to our viceroids, right, or sometimes, as I said, referred to as satellites. These are like viroids in that they're covalently closed, single-stranded, that's what the SS stands for, molecules capable of forming that structure I just showed you, right, of forming a loop and binding, interstrand binding with each other. But unlike viroids, they actually code for one or more gene products, right? So the ribosome can bind to a viceroid's RNA and make a protein from it. These guys get from one cell to another via a helper virus. In other words, they're inside of a virus. Viceroids, satellites are inside a virus. So we've already talked about the example that the hepatitis B virus is not, in fact, a totally different virus. Instead, it's a viceroid that infects the hepatitis B virus, which is, in fact, a virus. A DNA virus at that. And that's probably how they were able to figure it out, right? Because, as I said, currently, viruses are either DNA or RNA. They don't have both. So if you've got a DNA virus and you've got some RNA in there, that's a viceroid. That's a satellite, right? That's something else inside that virus. So prions or pyrons are sometimes just referred to as proteinaceous infectious particle or infectious proteins for short. I prefer that one. It's easier. Tells you a lot too, right? It's a protein that can cause infection. So some degenerative diseases in animals caused by infectious proteins are scrapie, bovine spongiform encephalopathy, commonly referred to as mad cow disease, Creutzfeldt-Jakob disease, and variant Creutzfeldt-Jakob disease, and CRU. So we're going to talk about the middle two, mad cow's disease and CJD. So in the 1990s, mad cow disease had reached epidemic proportions in England. Initially, it was spread because cows were fed meal made from all parts, including brain tissue, of infected cattle. So what they were doing is a cow would die, right? And they would basically feed that cow to the living cows. They made them cannibals. Are cows cannibals? No. When a cow dies naturally, they will leave it alone. They will not eat it. They're herbivores. They eat grass. Whatever possessed somebody to do this, I do not know. But they did. They thought, I guess, recycling, right? The problem with that is they obviously did not understand that that cow died of an infectious disease. Right? That they died of something that if some other organism ate it, including the exact same organism, a cow, they could get that infection. Understanding in prions and infectious proteins, this is a very relatively new area of science. There's still a lot of questions that haven't been answered. But definitely that it is a protein. And the next one, basically the human form of mad cow disease. So this same protein that will make cows go mad because it's a protein in the brain that deforms, it causes other proteins to also deform, we also have, right? We're animals, just like cows are animals. So Critchfield Jacob, there is a mutation that can happen, a change in that protein in a human being that causes this protein to convert all the other proteins like it into this inactive form of the protein. So this protein no longer does its job. And even worse, each one of them become renegades that convince everyone else to jump ship. So now you've got all these proteins not doing their job, instead convincing all their other friends not to do their job either. So the brain deteriorates. It becomes almost sponge-like, right? That's why it's referred to as spongiform, right? So that it becomes deteriorated, big holes in it like a sponge. So this can happen spontaneously, right? Somebody could have this mutation, have this defect, have this disease. The bad news is if you were to eat that person, particularly definitely their brain, you would acquire that protein, that mutated protein that convinces all the other proteins to also quit doing their job. So that's the virulent form, right? Where you ingest the infectious protein and get the disease that way. So how it came about is probably a mutation, right? Somebody's protein mutated, it went renegade. The bad news is if you eat that protein, your proteins will go renegade as well. So this is what happened to the poor damn cows, and why we wouldn't take any of their beef from England for the longest time too, until they cleaned up their frickin' mess, is because if we ate their cows, we would get that disease. And what's the really bad news? No effective treatment currently. We can't treat you. We can't save you. We just wait for you to die. Progressive degeneration of the brain. You can't live without the brain. Eventually, you die. Right. So, like I said, really new area of science. She asked if that mutation, like if you had it, could you pass it on to your children? And I would say, yeah, probably. So, what's that brain degenerative disease that you don't know you have until you're older? Parkinson's. So it might be something like Parkinson's. You know, why or how it activates and when. Like I said, there's still a lot of questions we don't know when it comes to this. So if you're into reading scientific papers, you know, there's plenty out there on this stuff. Active area of current research. The good news is that we do know about it now, so we can avoid it. Right? We're not going to eat each other. In the United States, we don't do that. You laughed, but there are countries, right, and that believe that if you eat your ancestors' brains, you get their knowledge. Right? We need to educate. Do not do that. It's not a good idea.
So they don't have a cure for Parkinson's disease?
Nope. No cure for Parkinson's either.
Well, there's have been donations specifically for people like Michael J. Fox. Have been donations for Parkinson's disease.
Right, but the thing is they have drugs and stuff that can kind of help with the symptoms. But they can't cure the main cause. And again, it's a genetic defect. But it's a genetic defect that doesn't turn into a disease state until you're older. So the bad news, too, is that people have babies, right? And you don't know you have this disease until you've already probably had a baby and passed it on to them. Right? So they can do genetic testing now for things like that, right? And so you can be informed and make those types of decisions. Do I want to potentially, if I have this, pass this on? And then, of course, some people don't want to know if they have it. They don't want to know if later on they're not going to be able to function anymore. So, yeah, there's a lot of, I guess. Yeah, because eventually, I mean, I forget exactly, it's definitely neurological. Parkinson's disease is neurological. I don't know the dysfunction, what it causes you to not be able to do anymore, specifically. But yeah, I mean, when things don't function over time, that's going to overwhelm the system and potentially kill you. Right? You can't do necessary things to continue life. So, do you still have a question in the back? No, right. Only if someone obviously, so if you ate somebody, right, only if they had this would you then get the disease. But I don't know about you, but I wouldn't particularly want to take the chance. Just saying. Right? So there are some religions, too, where they won't take other people's blood. Because blood can be highly infectious, right? There could be stuff in blood. I mean, there were people, you know, people that I knew I lost, that my family has lost, that got blood transfusions and it contained infectious agents before we could test for the presence of HIV, before we knew about hepatitis C, right? Before we could know. Like right now, again, what are they saying about the latest infectious problem we've got going on? The Zika virus, right? It gets into your blood. So, you know, if you were to donate blood, think about before we knew about it, before we could test for it. Someone could have got it from someone else's blood. The problem is, though, usually you're getting someone else's blood because you really need it. You're going to die without it. So it's like the kind of the risk that you take, right? So, does that make sense to you guys? All right, so moving on. So, viruses. So, the most widely accepted definition is an infectious agent having a single acellular, remember I will post for you.
I know what I want to do. This is blurry.
Oh.
I have my contacts and skills.
Oh, okay. I'll just ignore you. Her flashy phone is distracting. Okay. It's okay. I will ignore you. I watch all you guys. I'm a mom, you know, I am a teacher. I've got like that vision that goes everywhere, which can be distracting at times. Okay, so an infectious agent having a simple acellular organization, which means it's not a cell, right? Often just a protein coat and a nucleic acid genome. Lacking independent metabolism, right? It's not doing any chemical reactions on its own. Outside of a living cell, it's inactive. It's not doing anything. It's just there. Reproducing only within living host cells, right? It cannot copy itself without getting inside of a cell and using that cell's ribosomes, that cell's resources to copy itself. So, based on that definition, right, I don't consider them living. Most scientists don't consider them living. Others will argue. One of my colleagues here is a parasitologist, right? And she's like, but you know, some parasites can't replicate on their own either. I'm like, yeah, but it's a cell. That's my sticking point, right? It's an organism because it's a cell, right? It meets that criteria of being at least a cell. Granted, it can't replicate on its own. Okay, it's lazy. But it is a cell. A virus, on the other hand, is not a cell. Okay?
It's more like a parasite, like those mosquitoes.
Right. Yes and no. It's an infectious agent. Right? It can make us sick. It can take over. Right? It is definitely bad news, but it is not a living organism. It's not a cell. It doesn't replicate on its own. It doesn't do any metabolism on its own. So in science, where we have definitions, and these definitions really embody how we think about and interact, right, and categorize things. So, you know, that argument that I continually have with my parasitologist friend. Right? It's not a cell. So, acellular infectious agents, right, not a cell. People who study those are referred to as virologists, right? The study of viruses is referred to as virology. Our resident virologist here at Delgado is Marion Friedstadt, if any of you guys had her for biology class, right? Her background is virology. Specifically, she studied and still continues to study the polio virus. So she was away this summer on sabbatical and was working in a research lab that works on polio virus. So if viruses are what you think is cool and you want to talk to or find out resources, she's your go-to person for viruses, not me. I think they're cool though, right? But my level of understanding is the level that is in your textbook and that we need to be for this course. So we already kind of touched a little bit on the morphology, right? So there are different shapes that these virus protein coats can form. And viruses that are just that, just their nucleic acid and their protein coat, are commonly referred to as naked viruses. Because, especially as it applies to animals, humans, a lot of the viruses that infect us have an additional layer referred to as an envelope. So they're called envelope viruses. So, of course, those are the ones that we came in contact with the most. So when we found out about these naked viruses, that was it. They seemed naked to us. They're like, they're missing a layer, right? Why don't they have an envelope? They're naked. Make sense? Okay. So the nucleocapsid is just that. Nucleo referring to the genetic information. Capsid is the name given to the protein coat. So capsid is the name for the protein coat. And then the protein coat itself is made up of individual proteins. Those are called capsomeres. And the capsomeres themselves can form characteristic arrangements. So they can group in groups of 5 called pentamers, or they can group in groups of 6 called hexameres. when these proteins, right, these capsomeres form these pentamers and hexamers and form the capsid, this protein coat, they can form particular arrangements. They can be helical or isohedral, or they can be actually quite complex. So a viron refers to a single virus particle that contains its nucleic acid, it being DNA or RNA, enclosed in that protein coat. They may have additional layers, right? The most common additional layer, of course, is the envelope. And again, remember, they cannot reproduce independently of a living organism, a living cell. Therefore, they can't do cellular division like we're used to seeing in prokaryotes and eukaryotes, right? They're not doing binary fission or mitosis or meiosis. They can't. So a naked virus, again, is just nucleic acid, whether it be DNA or RNA, in a protein coat that we refer to as the capsid. Envelope viruses, just like a naked virus, have to have their nucleic acid, their capsid, their protein coat, but in addition, they'll typically have this layer we refer to as an envelope. And within that layer, there's usually these protein spikes. And these proteins stick out of the envelope, right? Because they stick out, they're kind of pointy. They're referred to as spikes. And they're very important for the attachment of the viruses to the cells that they infect. So their size is about 10 to 400 nanometers in diameter. So most of them really are too small to be seen with a light microscope. So you're not going to see them in microbiology lab or any of the other labs on campus. It wasn't until the advent of more high-powered magnifying microscopes like the electron microscope we're able to see structures of viruses. So all virins, again, have to have the nucleocapsid. They have to have their nucleic acid in their protein coat, known as the capsid. And then, as I said, some have an additional layer known as the envelope. Ones lacking an envelope that are just the nucleocapsid are commonly referred to as naked viruses. So the capsids, the protein coats themselves, are very large macromolecular structures. So they're not just a single protein, they're multiple proteins arranged in various arrangements to help protect that nucleic acid, but also help the transmission of it into the cells they infect. So they're made of protein subunits, referred to as protomers. So the two common arrangements, the first being a helical arrangement of the capsid. So here you have the tobacco mosaic virus. It's an RNA virus, so you have the RNA, but then attached to the RNA are the protomers, the individual pieces of proteins, but they form an overall arrangement, right, of spiraling up as a helical arrangement. That's a naked virus. That's it. It's just the nucleic acid in the protein coat. And this, of course, infects the tobacco plants. So it's actually a plant virus. Influenza virus. Right, looks like are we out of influenza season at this point, I hope?
No, I don't think it's
Next month? All right. Well, I escaped so far. Knock on wood. I got my vaccine. All right. So the influenza virus, its nucleocapsid is helical in arrangement, right? So as you see here inside, It looks very similar to the tobacco mosaic virus, right? In that you have RNA surrounded by a protein coat in a helical fashion. But that virus is an enveloped virus. So there's an additional layer, this envelope layer, that surrounds it. And this guy actually has more than one piece of RNA. It has a bunch of pieces of RNA surrounded in their protein coats in a helical fashion, all packaged up inside this envelope. Now, for the most part, envelope viruses, outwardly, right, if we just look at the outside, as you are seeing in this picture, they're pretty much spherical. They're not perfect circles right here because they're just kind of smushed into each other, right? But when we talk about the DNA, or the RNA in this case, the nucleic acid in its protein, that structure inside this virus is actually helical. Does that make sense to you guys? Most of the viruses that infect us have an envelope. Okay? And the reason for that will make sense as we talk about how they get into our cells. So another very common shape for the capsid is isohedral. And this is what that dengue virus is, that model that I have that you guys can print out and cut up. So if you look at it, it is a regular polyhedron, which means it has 20 equilateral faces, which means they're the same distance, and 12 vertices, 12 points. This is one of apparently nature's favorite shapes. And a lot of times they have these particular arrangements, these capsomeres. So with a ring or knob, units made up of 5 or 6 protomers. So if you have 5, it's referred to as pentamers or pentons, or hexons if there's 6. And so you can see in this picture right here, and you can see when you put together that model I gave you guys, you can see, I think that one has one pentamer it forms. If you print it out in color, you definitely will be able to see where I think 5 of the blue ones come together or something like that. It'll make sense when you fold it up. It's kind of cool. And then I have some in the lab as well, and I'm going to bring them to class on Tuesday. So, but you can see, here's the 5 for this one. Do you see the 5 there? It's forming the 5. Here, there are 5, and there's even a projection here. Here's 5 coming together. These are all the same protein around a point. And then off over here, you can see there's a grouping of 6. This is a hexamer. But the overall structure is this, right, isohedral. And a lot of viruses have this structure. So the I'm trying to think one of the picoviruses cause an infection for us. But adenovirus, this one could be transmitted to humans by mosquitoes, right? Cause a pretty serious respiratory infection. I don't know what the Zika virus is. I guess we need to find out what its shape. Last time I taught this class, we were talking about Ebola virus, man. It just never ends with these darn viruses. They're everywhere. So many viruses do not fit into the category of having helical or isohedral capsids, those protein coats. So some examples are the pox virus, which is actually quite unique, has all these strange additional layers. And then my favorite, the bacterial phage, or the viruses that infect bacteria. They look like little spaceships, don't they? They're really cool. Right? So you have the capsid head and the nucleic acid, but then you have all these other parts that literally allow it to land on the surface of the bacteria, and then it will inject its nucleic acid into the bacteria. And there's cool YouTube animations out there too showing this phenomenon of them landing on, attaching, right, like a foreign spaceship to the surface of the bacteria, and then compressing and injecting their nucleic acid into the poor bacteria. So we'll continue on on this on Tuesday.